to express my thoughts on a subject I feel is very relevant in today's times when health is the top priority on all our minds. So let's start by deconstructing these terms. We have two terminologies here, that is antibiotic and resistance. Let's keep the term resistance aside for a while now and go ahead with antibiotics. Now you all would have heard about antibiotics and I'm sure you have taken antibiotics in your lifetime, but let's just quickly see what they are when we speak in technical terms. See, antibiotics are again made up of two terminologies, anti and biotic. Anti is anything that is against, and biotic is a living system. So any compound or any drug that is against, acting against a living system, or in this case a microorganism, is called as an antibiotic. And when we say it is acting against a microorganism, it can either inhibit the growth of that organism or it can kill the organism completely. But either way, it is a compound that is acting against, anti, a living organism. That is why it is called as antibiotic. Now, there are different types of antibiotics and the range of antibiotics also are different. For example, we have antibiotics which have a wide range or a broad range, that is, they are able to infect a large group of organisms. And you also have antibiotics which are able to act against only a specific group of organisms or narrow spectrum of organisms. Those are called as narrow spectrum antibiotics. So the type of antibiotics are many based on their target organism, based on their target range. I think that is a topic we can discuss on some other day. But they also have different modes of action. So antibiotics could be acting on the cell wall synthesis of that particular bacteria or that particular microorganism. Now what I have shown you here are the different mechanisms that an antibiotic could act against or could act on in a bacterial cell. So if you see here, we have inhibition of cell wall synthesis. So the antibiotic could go and inhibit the cell wall while it is being synthesized, and cell wall is not there, the bacteria will die. Or it could go and disrupt the cell membrane of that particular bacterium, or it could act upon transcription and translation steps. Transcription and translation are part of protein synthesis. Or we could have antibiotics that are going and inhibiting nucleic acid synthesis, for example, DNA synthesis or RNA synthesis. Or we can also have anti-metabolites. Anti-metabolites are those antibiotics which are going and inhibiting a particular biochemical pathway. So I have a pathway wherein I have synthesis of B from A. So I have an enzyme that is acting on that pathway. I could design an antibiotic that is going and acting upon that enzyme and stopping that pathway, inhibiting that pathway. These are the different target areas, or these are the different places an antibiotic could act on in an organism. And that's how it is able to inhibit the growth of that organism or kill that organism. This is how an antibiotic works. Now, earlier, an antibiotic was restricted to a compound that is synthesized by a microorganism and is acting against another microorganism. But now, the terminology has been widened to include any compound either synthesized by a microorganism or being chemically made in the lab which is acting against another microorganism. So as long as it is acting upon a microorganism, we are now calling it as an antibiotic. So initially it was restricted to the compounds that are synthesized by one microorganism and are acting against another microorganism. I am sure all of you would have read in your lower classes or lower grades about the first antibiotic that was discovered by Sir Alexander Fleming by mistake or serendipitously. He had discovered the penicillin from the fungus Penicillium. That was the first antibiotic to be discovered, and after which we have several antibiotics that have been discovered from fungi and from actinomycetes or even bacteria, which are active against other groups of organisms. So that is why that was the original definition of antibiotics. But like I told you, the definition has been widened now. So as far as antibiotics are concerned, it's a huge topic in itself. We'll discuss about it later. Let us come to the topic of today, that is resistance. Now, resistance as such is something that people are not happy about. When the Indians resisted British were not happy about it. Or when the farm workers resisting, or when they showed resistance against the landlords were not happy. Or when college students show resistance against the college management and the teachers, they are not happy. So resistance is something when we are going against a system or when we are going against something, that is called as resistance in plain English. Now, what is the problem here? The problem here is antimicrobial resistance, or when the microorganisms are resisting antibiotics. So it can be called as antibiotic resistance or even antimicrobial resistance, which is abbreviated as AMR. Now, AMR is when we have the organism, microorganism resisting the antibiotic. So I put in a lot of effort, I create an antibiotic, or I put in a lot of effort and discover an antibiotic from a microorganism. I work upon it for years together, make sure it has no side effects, bring it to a dosage that is apt for a human being, and then convert it into a form that can be taken orally. I put in a lot of work in it. And then when I finally give it to the person, in a few years, my microorganism becomes resistant to it. It does not, the drug does not have any effect on the organism. That is called as antimicrobial resistance, and that is the problem over here. You would have read about antimicrobial resistance in books, and if you have attended seminars, you would have definitely heard about AMR and antimicrobial resistance. But I think it will be shown here in a very simple manner by these two favorite cartoon characters, Tom and Jerry. Let's see what they have to show. So we have Tom, who is the human being who is designing an antibiotic, and Tom is being very smart and coming up with an antibiotic. He's testing the antibiotic on a particular organism. So here we have the new antibiotic, and he finds that it is very, very effective because the organism is dying. This is what he has done in the lab. Okay, so the organism has taken a small quantity of antibiotic and is dead. Very happy with it. The human being is now going to test it on the actual organism which is causing all of the diseases. But if you see here, the organism was supposed to take only a bit of the antibiotic. Instead, the organism drinks up the whole antibiotic. So this is the bacteria, our pathogen. The pathogen is now going to drink up the entire antibiotic, or it is going to take a large dose of that antibiotic. Let's see what happens after that. So assuming it's dead, we continue with our activities. But instead, what happens is now the pathogen or the bacteria becomes extra strong. So if you can see here, now we have the antibiotic resistant bacteria. So what antibiotic we had created is now actually of no use. So we create a new antibiotic, for example, ampicillin, but that has no effect on our bacteria. Our bacteria has become too strong for it. So we go ahead with the next antibiotic. Ampicillin didn't work, no problem. We'll go for cephalosporin. Not just one cephalosporin, different generations of cephalosporin, better versions of cephalosporin. That is also not effective. Then we go for gentamicin. That is also not effective. We tried different antibiotics. Now we have gone for tetracycline, a higher version of antibiotic. But look at our bacteria, how it is pulling out diseases. The antibiotic has absolutely no effect on the bacteria. And in this way, if you see, the bacteria has taken care of the human being, or in other words, the bacteria has surpassed the human being. When we thought we were being smart, the bacteria or the pathogen has outsmarted us. This exactly is the problem with antimicrobial resistance. This is the actual crux or meaning of antimicrobial resistance. Now, how does the organism get resistant? So, the organism has different mechanisms by which it is able to get resistant to an antibiotic. So, initially, the antibiotic was able to work on the organism. When I designed it, it was killing my organism, or when I designed it, it was inhibiting the growth of the organism. But now what's happening? It can adapt any one of these mechanisms that are shown over here, or it can adapt a combination of these mechanisms. For example, the organism could show decreased uptake to the antibiotic. It is not letting the antibiotic in at all. It is not allowing the antibiotic to enter. That could be one of the mechanisms which has been shown over here. So decreased uptake could be one. Not letting the antibiotic enter. Somehow the antibiotic enters inside, but there can be the next mechanism wherein the organism is having efflux mechanism or pumping out the antibiotic. It is kicking the antibiotic out. Somehow the antibiotic has come in, but the organism does not let it stay in. It is going to push it out or pump it out. Or the antibiotic can do modification by producing some enzymes. Now the modification can happen in two places. Either the organism can modify the antibiotic itself. So it does not allow the antibiotic to function. If you can see here, once the antibiotic has come inside, my organism has changed something in the antibiotic and now the antibiotic is not functional anymore. Or it can change the target. Now the antibiotic was supposed to go and act upon a particular site on the bacterial cell, but what does my bacteria do, or what does my pathogen do? The pathogen changes the site itself. So even though the antibiotic is there in the vicinity, it is unable to identify the target because the target has been modified. Or it can go for alternate pathways. Alternate pathways is wherein I had production of A to B by using a particular enzyme. Human being thinking we are very smart, we block that enzyme. What did our bacteria do? Okay, you don't want me to produce this enzyme? It's fine. I'll produce B by a different pathway. So it comes up with a different pathway and produces the product anyhow. So these are some of the mechanisms by which the bacteria or the pathogen are able to gain resistance. That means how much of an antibiotic we give, if it has any one of these pathways also, it is not going to be effective. I can give kilograms of antibiotic, but my antibiotic is not going to be effective or it is not going to work upon that particular pathogen. This is the problem that is occurring. So we know we have antibiotic resistant bacteria. We know we have antibiotic resistant organisms. The reason they are resistant to these antibiotics is due to one or a combination of these mechanisms. So we now know what antibiotic resistance is. We know how the antibiotic resistance is occurring. These are the mechanisms by which they occur. But why is it a big problem? Why am I talking to you about this today? Or what is its impact on public health? Now what happens is, when a person is suffering from a disease and I have to, being a doctor, if I have to administer antibiotics to that person, I need to know what that person is suffering from. So I'll be doing a test and I'll be finding out what the organism is. That organism will be susceptible to, or it will be sensitive to, a few antibiotics. So what do I do as a doctor? I prescribe any one of those antibiotics in a particular dose and ask that person to take the antibiotic for a particular number of days or to complete a course. Now, if that person does not complete the course, or suppose I had prescribed the antibiotics for 5 days, but the person takes it for 2 days or 3 days and sees that, oh, I don't have any symptoms anymore, I can stop the antibiotics, what happens over there inside the body? I would have had several organisms growing, the antibiotics came and attacked it, and most of the organisms died. But do you all remember the rule, survival of the fittest? That happens here as well. So though most of the organisms died, we had a few microorganisms, we have a few bacteria or a few pathogens which survive. And they survive, that means they are able to resist that antibiotic. So most of the organisms have died, but a few in that population survived and now they have become resistant to that antibiotic. So I was supposed to take the medicine for 5 days, but I took it only for 3 days. At the end of third day, I felt I'm fine and I stopped the medicine. What is happening there? After the third day, the fourth, fifth, sixth day, that resistant microorganism develops. That resistant microorganism starts multiplying. It creates more of itself inside my body and I get my infection back. And this time, will the antibiotic work? No, because my organism has already become resistant to it. So now the doctor has to find out what is the antibiotic that I have to be given. He may have to give a higher dose of that particular antibiotic, and this process keeps going on. So the main reason why we are talking about this, or the main impact on public health, is that if we don't take this problem in check, if we don't check this problem now, day by day or with time, we won't have any solutions at all for any of the diseases, because every solution we come up with, every antibiotic or drug we come up with, I have the organism getting resistant to that particular drug. So what happens over there? When the organism gets resistance, I don't have any drugs with me. By the time I build another drug and come, my organism would have become resistant again. And people will have infections which we will not be able to take care of, or which we will not be able to treat. So that is the reason why I chose this topic, or why I feel it is required for all of us, especially in the field of biology, to know this. If you are not at the end of prescribing an antibiotic, at least if you are at the receiving end, at least when you are given an antibiotic or your loved ones are given an antibiotic, you need to be careful that you complete the course. You need to be careful not to take a very high dosage of antibiotic. That is why you need to know about this. So how or when exactly does an organism gain resistance? One is when we don't complete the course of antibiotic. The other situation can be you take an antibiotic unnecessarily. I have a headache. I could just stay with the headache for a while, drink a lot of water, rest. I might be relieved of the headache, but I don't do that. I look for a shortcut way. I start an antibiotic. When we start self-prescription, that is a big problem. We will end up taking antibiotics unnecessarily. So the unnecessary use or the non-judicious use of antibiotics is a big problem or is a big reason why we have this problem today. Or over the counter sales of antibiotics. I go to the shop and I tell the doctor has told me to take this particular antibiotic. The person, the pharmacist there doesn't see my prescription and just hands me the antibiotic. That is also a problem. I should not be allowed to go to the counter and ask for any antibiotic without a doctor's prescription, because you may not need the antibiotic for that infection. That infection might be very mild. You can let it go, especially when we talk about common cold or any flu infection, these or any viral infection. These are infections which are caused by viruses, and viruses don't respond to antibiotics. Viruses respond only to antiviral agents. That's a very few of them. So if we take an antibiotic for a viral infection, what are we doing over there? We are making the other bacteria in our body resistant to that particular antibiotic. And when we need that antibiotic, when we actually need that antibiotic, the bacteria has already become resistant to it. It's not effective on it anymore. So that is a big challenge or a problem over here. In fact, it has been told by CDC, that is Center for Disease Control, that this is one of the biggest threats world over. It is not just in India, worldwide, this is one of the biggest threats. We do have life-threatening infections, but AMR or antimicrobial resistance is a big issue. In fact, you can read the complete AMR report that is released every year on the CDC website. They haven't released it for 2020 yet. What I have over here is the AMR report for 2019. So based on that report, in that report, they have listed out the different organisms to which we are already becoming resistant. We already have those organisms which are resistant to antibiotics. And what they predict is that there are, or what they say is that there are more than 2.8 million cases of antibiotic resistant diseases that are occurring in US every year. In fact, there's also a report, a research article where they say that there are 2 million deaths which are projected to happen in India due to antimicrobial resistant organisms by the year 2050. This 2 million deaths which India is predicted to have is part of the 10 million that is predicted to happen by 2050 worldwide. So out of the 10 million deaths that can happen worldwide, 2 million are in India itself. In fact, India is one of the leading users of antibiotics because of all the reasons that I just told you. So we go over to the shop and we are allowed to buy a lot of antibiotics over the counter, which should not be, which are actually not allowed to be given like that, which have to be given only with a doctor's prescription. But we have been doing it and we continue to do so. We take it as and when we want. When the symptoms stop, we stop taking it. When again the symptoms arise, I start taking it. What are we doing over there? We are making the organism more and more resistant. The organism is evolving faster than us. The organism's evolution makes sure that it is able to survive that particular antibiotic. Another reason which has been quoted is that the poor sanitation conditions in India is also one of the leading reasons why we have been predicted to have these many deaths by the year 2050. Because in India, the environmental conditions are such that there might be pharma companies or even hospitals and other medical and research centers which are releasing organisms into the environment without treatment, without proper treatment. Now these organisms, some of them might be resistant. They may transfer the resistance to other organisms in the population. That is also happening. I can have transfer of resistance to a gene, or in the form of a plasmid, I can have transfer of resistance from one organism to another. So if I don't kill the organisms in my lab before I send them out, I'm very much transferring resistance to another organism. There could be some organism in my lab which I'm working with, which is resistant to an antibiotic. I'm sending it out into the open, and that could go and pass on the resistance to many more such organisms. That is also one problem. If you see over here, I have displayed a tabular column which is showing the different organisms that are resistant to antibiotics. So if you see here, the first one is penicillin, and penicillin was discovered in 1941. It was released into the market, not discovered. It was discovered in 1929, but the consumable form of penicillin was released in 1941. But if you see here, you can see that the year in which it was already the penicillin resistant organisms were identified is 1942. By the time we were able to release the antibiotics, we already had organisms which were resistant to it. Initially, they were susceptible. The strain became resistant. So we have this classical example of penicillin resistant Staphylococcus aureus. Now this Staphylococcus aureus was initially susceptible or sensitive to penicillin. That this was the organism through which we discovered penicillin. But because it became resistant to penicillin, we had to come up with another antibiotic. Staphylococcus aureus causes a lot of skin infections and other infections. So we needed to come up with an antibiotic. Because penicillin wasn't working, we modified the penicillin and we got another version, that is methicillin. If you can see over here, we have methicillin that was discovered. Now this methicillin, by the time we got this methicillin into the market, we also had methicillin resistant Staphylococcus aureus. Now methicillin resistant Staphylococcus aureus is also abbreviated as MRSA, and this is one of the leading causes of hospital bone infections. Now methicillin resistant Staphylococcus aureus was resistant to penicillin and methicillin. So we had to come up with something new, some new antibiotic to beat it. We came up with vancomycin, but we had vancomycin resistant Staphylococcus aureus. Then we came up with daptomycin. If you see the second last row, we have daptomycin, but the organism became resistant to daptomycin as well. By 2004, the staph or Staphylococcus aureus became resistant to even daptomycin. Now, what happens when people get such infections? If initially I had Staphylococcus aureus, I could have administered penicillin. It's not working. With this strain, it's not working. So then I have to go for methicillin. Again, with that strain, methicillin is not working. We went for vancomycin. It's not working. We went for daptomycin. Now we have strains which are not even responding to daptomycin. So what do we do if a person is infected with such a strain? How do we treat that person? How do we make sure that the infection is completely removed? That is the problem here. That is the challenge. We don't have any more antibiotics. The organisms are growing resistant to these antibiotics day by day due to the improper usage, due to the overuse, due to the wrong use, excessive use. Too much of antibiotic is being given. Very high dose of antibiotic is being prescribed by doctors. Even some doctors are at fault in this. Some of the medical personnel will give a high dose of antibiotic in the first round itself, where a very less dose of antibiotic could have worked. They are prescribing a higher dose. What is happening over there? The organism is getting resistant because you keep giving it a higher dose. It will get used to the higher dose. So these are some of the issues that are there with antimicrobial resistance. And as biology students, what is our role over here is to make sure that we don't contribute to it. We should make sure that we don't take antibiotics unnecessarily. Where there is need to take antibiotics, you have to take it. For example, if you see here, these are posters that are released by CDC. There are treatments or there are infections where you need to take antibiotics. There is no other way out. For example, if there is sepsis, for treatment of sepsis, or for post-surgical infections, or if there are some chronic diseases, you need to take antibiotics. Or if there are patients who have undergone organ transplantation or dialysis, or if they are cancer patients, they'll have chemotherapy, you have to administer antibiotics. So because their immune system is already suppressed, you need to make sure they don't have any other infection and you have to administer antibiotics. But apart from these, unnecessarily taking antibiotics for common infections, for mild infections should be avoided. Unless and until the doctor feels, unless and until the medical personnel feels that yes, this infection needs an antibiotic to be rectified, we should avoid the usage. Also, we shouldn't do that. I'll take this antibiotic now, I'll take it for a few days. If the symptoms go, I'll keep it, I'll store it. If the symptoms come back, I'll take it. No, that shouldn't be done. If a course has been prescribed for 3 days, take it for 3 days and stop. Don't take any more, don't take any less. If you feel that the antibiotic is not working, go to the doctor. They may change the antibiotic or they may change the dosage for you. Don't do it by yourself. Don't go for self-prescription. Don't go for self-treatment. Always make sure that the person who knows what has to be done is involved or is in charge, and they are the ones who treat you. Otherwise, antibiotic resistance can not only harm people, it can also cause transfer of many organisms from animals to human beings, and it can also cause issues in the environment where we have antibiotic resistant organisms in the environment and we'll not know how to deal with them. So these are some of the issues of antibiotic resistance or antimicrobial resistance as it is commonly called, which are very, very important for a biology student to understand and to be sensitive about it. If we take care, definitely this problem can be reduced. When we work in the labs, we need to make sure that we don't add on to the problem. We should make sure that we decontaminate the organism, kill the organism there, and then only send it out so that we don't add on to the problem of drug resistance or antimicrobial resistance, and we are not involved in creating super bugs, bugs which cannot be treated by any antibiotic available to us. I hope I have been able to throw some light on this very, very pressing issue, very, very serious threat that is there in the world. If you have any further doubts or queries, you can always contact me at the email ID given below. Thank you for giving me this opportunity. Thank you, one and all.